Hello everyone, this is bioscience lab. Do you want to know how students do experiments in this lab? Okay, come in with me and I will show how they do their experiments. Come on.
This video will introduce you to a specific pipette, pipetteman. There are 5 different kinds of pipettes in our laboratory, each with its own kind of tip, and each pipette is designed to transport a specific range of volume. The tip in a large blue box fits the P1000. The tip in medium-sized box fits the P100. The tip in the smallest box fits the P20 and P10. To achieve the greatest accuracy, it is best to select the smaller pipette that can handle the volume you wish to transfer. You should always wear a lab coat and use gloves. If the pipette tip touches any surface or another sample, then change the tip to a clean one. You should keep your pipette mostly vertical to prevent liquids from running up the inside of the shaft of the pipette and into the dispenser. Press the pipette shaft down onto a new tip with a rotating motion so the point is well sealed to keep it from leaking. When finished, you can press down the eject button to eject the tip. Be careful not to stab the pipette into anything, since it will likely damage the pipette and impair its accuracy. A digital volume indicator has 3 digits. We can read the volume of liquid that we are going to suck up in the pipette. Different types of pipettes read differently. If it is P1000, it indicates that the max volume of this pipette is 1000 microliters. So this is 1000 microliters. If it is P200, it indicates 100 microliters. If it is P20, it indicates 10 microliters. To set the volume of liquid to be dispensed, adjust the volume knob. Hold the body of the pipette in one hand and use the other hand to rotate the thumb wheel or the push button volume setting. Always finish the setting with a brief clockwise motion to minimize the error caused by mechanical backlash. When decreasing the volume setting, turn the thumb wheel slowly until the desired setting is displayed. Be careful not to overshoot the required position, or you will have to refill the pipette. When filling, increase the volume setting. Rotate the thumb wheel approximately 1/3 of a turn above the desired setting, and then slowly turn back to decrease the volume until you reach the desired setting. The push button is first pressed to the first stop, and this expels a volume of air equal to the selected volume of liquid. Aspirate the sample and release the push button slowly to fill the pipette, and not have the sample rush into the shaft and cause contamination. Notice the position of the pipette tip as it is immersed in the sample. If it is too deep, it may form extra droplets outside the tip, and they will be deposited along with your calibrated sample. If the tip is not deep enough, then vortexing will occur in the liquid, and your pipette will suck up some air instead of the selected volume. If this happens, dispense the sample and try again by pressing the push button to the second stop in order to expel the incorrect sample in the pipette. Only release the push button after the tip has been removed from the sample. Change the tip after each sample. After dispensing, you need to store the instrument vertically and especially avoid a cross-contamination of the tip.